Right. So you should be able to see the slides now. So so this session is discussing maternal pediatric precision therapeutics data model knowledge and research coordination center, which we call simply the DMKRCC. So just to introduce a little bit of the, Aaron gave a little bit of our information about it, but to give a little bit of an overview of the setup for the DMKRCC, Langley and I serve as the PIs for the center, and then so it's based out of both Indiana University and Ohio State University. We have 5 different cores. The logistics core and outreach and dissemination core do what they say they do. Provide oversight for the Imprint DMKRCC as well as the Imprint Hub Steering Committee, and also then help support outreach and education through the website, social media, and other aspects. Then there are 3 kind of research cores. As Aaron mentioned, the knowledge base and portal core is a major component of the Imprint Hub, with led by Langley out of Ohio State, and will develop the ontology and knowledge base to collect information pertaining to pharmacokinetics and pharmacodynamics in maternal and pediatric populations. Rob Bees and I lead the pharmacometrics and clinical trial design core. So it goes both sites within Indiana University and the University at Buffalo. And then the real world evidence core has been led by Anita Mendoko and Christopher Bartlett. And Sean Granis will be taking over that as Anita has moved to Cincinnati, although she will still be involved in the core. So we'll give some information about each of these cores throughout the talk today. But we also have numerous co-investigators from Indiana University, Regan Street Institute, Ohio State University, and the University at Buffalo, as well as Purdue University, who help participate and get this work accomplished, as well as the numerous students, postdocs, and staff members who are too numerous to mention, and I'm sure I would miss one, but who are the ones who actually do all the work for this grant. And then we have an internal advisory board with experts from in pediatrics and neonatology, Wade Clapp and Laura Hanneline from here at Indiana University, David Haas, who's the vice chair for research and OBGYN at IU, Todd Score, the director of the Indiana Institute for Personalized Medicine, Andy Dahlem is the former division chief of clinical pharmacology at IU, Titus Schuyler, who's a professor of biomedical informatics at IU and Regan Street Institute. Mitch Phelps has expertise in pharmacometrics from Ohio State University, and Bill Jusko, the distinguished professor of pharmaceutical sciences at University at Buffalo. So we'll hear a little bit more in a few minutes about our outreach and education core, but this really largely provides monthly educational webinars, helps support things like this annual meeting, also supports the website at imprint.org and our social media sites on Twitter, Facebook, and LinkedIn. Again, I'll provide some additional information on our activities from the Outreach and Education Subcommittee this afternoon that will discuss more of our work here. And then the logistics core also has the opportunity to provide research support and funding to help expand and catalyze research in maternal and pediatric therapeutics. We have an opportunity pool funding that can help address emergent needs and leverage novel technologies to support research. And we will discuss this more tomorrow morning at our session for opportunity and support pool funding. So join us then to hear more about this. With that, I will turn the talk over to Dr. Langley from Ohio State University.
Thank you, Sarah. So this is the knowledge base and portal core. Next slide, please. Yeah, I'm gonna start from early version of the grant where the core has four aims. The first aim is to develop ontology for pediatric and maternal patient population and drug studies. Aim two is developing machine learning AI tools for natural language processing because the goal is trying to use NLP to help the data collection and the data accumulation. Aim three is dedicated for the knowledge base development itself. And we do have a team of data curators to manually curate the data from paper which is relevant to pediatric and maternal patient studies. Finally, we're developing APIs, allow investigators to come in and query the data knowledge base. Next slide, please. So this is a really kind of an overview of maternal knowledge base ontology. For the maternal population, we have pregnancy population. Sorry, Sarah. So when Sarah trying to get the slides back, let me continue talking. So basically, the ontology itself, though, is trying to divide up the maternal population between the pregnancy and the postpartum populations. Will be interesting for both physiology and non-physiology parameters, and we'll also divide those studies into pharmacokinetic study, which focusing on the drug exposure studies. And we also do pharmacodynamics, clinical trials, pharmacoepidemiology, plus omics study. Those type of studies not necessarily are totally independent. There are some studies cover both PK and PD, and some clinical trials are going to include PK and PD components as well. Okay. And there are sub-studies, some are more clinical driven, and some of the PK study are in vitro cell-based study. For some of the epidemiology study, there could also be phase 3, maybe even a phase 4 study as well. Okay. We do consider in silico study too, because those studies are going to help building up the PPK models and the other system pharmacology model too. And in the ontology, we also spend quite a bit of time to look into the specimen of the samples, disease elements, social determinants, variables, and other study variables. Okay, and one important thing about the annotation scheme of the ontology for maternal population is about the timing. We're talking about it in the pregnancy. We're talking about trimester number 1, 2, 3. And we also spend a lot of time thinking about the metabolism and the transporter pharmacokinetics related mechanisms. And we're going to curate those in vitro data elements if we can find them. Okay. So this is a kind of an overview of maternal knowledge-based ontology. Next slide, please. And similarly, in the pediatric knowledge-based ontology, we have almost the same framework. The only difference, though, is the subpopulation definition. We're talking about fetal, preterm, neonates, infant, and a bigger 1 year old. So that's the major difference between maternal ontology. Next slide, please. Then this slide tells you how do we do the data curation. So technically, we're going to spend a lot of effort going back to literature, PubMed abstract, identify the relevant paper. So that first phase is called a text classification. And once we identify the PDF file, we're going to convert it into text file, and using name entity recognition, which is focusing on different parameters, including PK/PD and the epidemiology studies, and we're gonna have labeled attacks. And with labeled attacks, we have the menu creating through creators that build the data into the database. Okay. In terms of natural language processing technology, we're using active learning and deep learning strategy to try to optimize text classification performance, because that very first step is really controlling the workload of the whole data creation workload. Next slide, please. All right, so this is a kind of screenshots of data curation tool for PK parameters. So this is really the data collection step where we gather the data into the system. So largely this is really for back-end data collection purpose. So this is used by data curators, not for general usage. So that's what we do in a PK study. Next slide, please. And the data sources wise, we focus on not only PubMed, but also looking at drug labels, clinicaltrial.gov, drug bank, FAIRS, PharmGKB, and other existing ontology database in the public domain raw data set. Next slide, please. And the following several slides are the knowledge portal where we recently just developed it. It right now has the opioid drug classes. So we did the PK study, parameter data collection as well as pharmacoepidemiology study section. And this is going to overview of all the studies. The first figure is how many pharmacokinetic studies by different study type. And then the second one is what's the pharmacoepidemiology studies by study type, okay, PE and the CT. And the bottom two figures are talking about the frequency of PK study in a different patient population. We're talking about maternal population from pediatric population from fetal all the way to the infant to the bigger than 1 year old. And then pregnant women, we have pregnancy, lactation, then the 3 stage of pregnancy as well. So you obviously see the different distribution, and the y-axis is the number of publications. Okay, so that's is overview of this portal. Next slide, please. And this is really PK parameter portal where you can go in and seek all the PK related studies and parameters from different papers. Okay, the first column is a PubMed, then there's drug names, specimen samples, concentration, so on and so forth. Next slide, please. Oh, hold on. Can you go back a little bit, Sarah? All right. So we're gonna have another 1 hour session tomorrow, focusing on the knowledge-based development portal, and has more interactive process where our team member Aditi and Lei going to show you the web portal, and as a training. I think largely if you have more questions, definitely join us tomorrow. Then we can talk more about this knowledge portal we have developed so far. Next slide, please. So this is a true hero. Aditi and Lei are 2 people behind this. Lei is the one actually designed the web portal and data curation tools, and Aditi is the one actually developed ontology, really define interface of those data portal. And Shijun Zhang is the primary data curator, and we have 2 students participating in the machine learning AI technology behind it. So that's what they have accomplished. All right, so that's my slides.
All right, thanks, Lang. So now we'll turn over to discussing the pharmacometrics and clinical trial design core. Again, this core is run by myself and Rob Bees from University at Buffalo. So I'll give a little bit of background and then turn it over to Rob for further discussion. So one of the goals we have as part of the pharmacometric core is to support the knowledge base and portal core to catalog maternal and pediatric PK and PD models. So what we will be doing is supporting a similar database to what Lang's work is doing, but this is more focused on identifying the types of models, specifically PBPK type models, as well as the population PK models, whereas the knowledge base and portal core information shown previously will be more of the descriptive non-compartmental analyses. So we'll break it down by drug name as well as population, and then give information on the type of model, as well as some information about each of the studies or publications that are made, the model structure or software that's being used to create the model, whether code's available or data's availability, and then some other remarks about this. Then evaluating all those, we'll take a look at best practices approaches for developing maternal and pediatric PK models, as well as supporting pharmacokinetic and pharmacodynamic study design in maternal and pediatric populations. This will incorporate literature review to develop the adult model and verifying that using either literature or other observed data. And then adjusting system parameters to predict what the pharmacokinetics will look like in a maternal or pediatric special population, and then doing study simulations and hopefully showing some observed data for those where we have observed data to help validate our models. And we can see here from one of our studies that shows this validation of a maternal pharmacokinetic model for betamethasone, the concentration time curve overlaid with the observed data from 2 different studies. We've done several studies here at Indiana University recently that are being analyzed using both population pharmacokinetics and PBPK modeling. The IMPROVE trial was a randomized, triple-blinded, placebo-controlled study of vaginal versus buccal administered misoprostol for induction of labor in 300 uncomplicated singleton-term pregnancies. They were dosed based on our clinical guidelines at either 25 micrograms misoprostol for 1 hour, or sorry, for 1 dose, and then every 6 hours after that, they received 50 micrograms misoprostol as clinically indicated up to 48 hours. You can see here that our clinical study in these 300 women showed that the vaginal administration of misoprostol led to a shorter time to delivery than buccal administration. As part of this study, we collected pharmacokinetic samples during the first 2 dosing intervals, or first 8 hours, from a subset of about 50 women, 24 buccal and 23 vaginal administered drug. And you can see from just a plot of the concentration versus time data that the vaginal dosing dose drug in the blue line had higher concentrations than drug that was administered buccally, shown by the red line here. We conducted population pharmacokinetics analyses and found that the bioavailability of vaginal misoprostol was about 2.3-fold higher than that of buccal misoprostol, and which explains the higher AUC and exposure observed by vaginal administration, and may in part explain why there is a shorter time to delivery following vaginal administration. We've also conducted a study on gabapentin in the immediate postpartum period. This is in women undergoing C-section who received gabapentin 300 milligrams orally prior to surgery, and then every 6 hours for up to 48 hours as part of an ERAS protocol at one of our institutions. And you can see we had 21 women participate in this study, where we collected plasma and breast milk samples, as well as some infant samples during a single dosing interval in these women. This shows some preliminary information from our modeling work on this, where we found, the goal of the study was to estimate breast milk concentration and relative dose to the infant. And we found there is gabapentin excreted in breast milk. It's a relatively low concentration during the relative infant dose over the first 24 hours postpartum is about 0.5%, and it's about 3% or 350 micrograms per kilogram per day during the second 24 hours, so from 24 to 48 hours of life based on our calculations. We did look at two different models to compare which may be better in identifying breast milk excretion and properties, and they both gave very similar results. So we also have done a study of pediatric pharmacokinetics of methadone in a post-op setting. This is work by Cynthia Sabenstein, who's one of our collaborators and is now at Pittsburgh. In these children, largely adolescents undergoing spinal surgeries, they received methadone intraoperatively and then every 12 hours while inpatient. And what we found in this study, looking at both R and S isomers of methadone, was that there's an effect of the alpha-1 acid glycoprotein concentrations in the plasma, as well as a SNP in the alpha-1 acid glycoprotein gene on exposure for both R and S methadone. And then CYP2B6 and CYP3A4 variants and phenotype play a role in the clearance of both R and S methadone. As you can see here, CYP2B6 phenotype has a major effect in poor metabolizers and rapid metabolizers. Now I will turn the talk over to Dr. Rob Bees, who's the my co-PI at the University of Buffalo.
Thank you, Sarah. So as Sarah indicated, I'm the co-director of the PCTDC with her, and I work also closely here at UB with Dr. Dhaval Shah on this, and the fellow that's been doing quite a bit of the work in this area is Yifan Yu, a PhD candidate in the laboratory. Next slide, please. So in terms of collaborative project support, so one of the great opportunities we've had is to be able to contribute to study ideas. So we were approached by investigators regarding applying optimal design and simulation estimation strategy evaluations for different study designs for a PK study assessing injectable cabotegravir and rilpivirine in virally suppressed women with HIV during pregnancy and postpartum. And this was to assess a trial design that would look at both monthly and Q2 monthly cabotegravir and rilpivirine long-acting formulations, in particular in the second and third trimester of pregnancy. As I mentioned, we used a population optimal design, and we had actually had a prior population PK model that was developed from the HPTN 077 study. And then refine that with a simulation estimation strategy to assess the trial design in more detail. If I get the next slide, please, Sarah. Thanks. So this is showing the optimal design results. So initially, we were able to take this population PK model, and we were able to evaluate the second and third trimesters in terms of what samples would give us the best information with respect to estimating the PK parameters, in particular the absorption rate and the clearance. And so on the left, what you see are the original design where we had actually intermediate samples to try to get both the Cmax and the trough, and the red line is the every 4 weeks group, and the blue line represents the Q8 week model. On the right hand side, you see the simulated virtual individuals. You can see that there's quite a wide range of potential exposures, and then the sampling times superimposed over this. And you can see the differences in the concentrations quite starkly in terms of the distributions for the Q4 week versus the Q8 week distribution. So if I get the next slide, please. So the population optimal design actually resulted in some further refinement in terms of changing that interdose interval timing for the sampling that would give us those optimal points. Although by using we use the POP-ED implementation from Uppsala, I believe that's Andy Hooker's software implementation. We brought this back to the investigators. So, and they said, well, but is it practical really to try to do these specific detailed interdose intervals when we can do additional samples, in particular in the Q4 week group, is going to be in the second and in the third trimester, when the patient is going to come back on a weekly basis for clinical visits. So that we'd have the opportunity to take an opportunistic sample sometime in that interval. So if we get the next slide, we then moved on to a simulation estimation paradigm. So using our population model, we simulated the design where we've got trough samples, and this is showing the Q4 week group. And it's showing the Q4 week group because we were initially going to update the parameters and assess the expected exposure in that Q8 week group before the arm for the Q8 week group is open. And by using the simulation estimation window strategy, we actually could incorporate the plus or minus 7-day window that is expected with respect to when a typical clinic visit may occur when the patient comes in for redosing. And then you'll see that in the third trimester, we added a sampling window that was at least a week from the dose visit, and gave a plus or minus 2-day time window, and we were able to demonstrate that one could get reasonably good estimates for the key parameters using this design. So we get the next slide, please. So again, actually I've already sort of discussed this, but we then adapted this model. So there's some controversy about what the expected changes in pregnancy with cabotegravir. So one group, I believe the Liverpool group, published a SimCip-based, published they have a pregnancy model and simulated. There was really no expected difference in exposure. So we took an existing model for dolutegravir that was published by Anders Dahlman. And given the similarities between dolutegravir and cabotegravir, we adapted it for cabotegravir by putting in the cabotegravir specific compound characteristics, and then simulated the Q4 week group again. So if I get the next slide. And so this is just showing the model structure. We did this in Open Systems Pharmacology. We now have this in MRGSolve, and this will be one of the models that ends up in the repository in terms of being accessible for anyone who would like to use it, at least in the MRGSolve form. And then just we show the drug transfer that was equations that were used that were based on the publication from Anders Dahlman. So we actually can simulate also in the fetus, but I'm going to focus on the maternal exposure to start with. So if we get the next slide. So what we see here on the left-hand panel is the non-pregnant simulation starting at 0. So these patients are either going to be continuing. So one of the things we learned is these patients are going to be continuing on the long-acting dose or actually switching. So they'll be on some oral therapy, have an oral lead-in and continue. But just for the sake of illustration, This is in the non-pregnant individual, you see that you're well in excess of the 8 times PAIC90 based on the dose proposed for this study. And in pregnant individuals, you can see that once you get closer to steady state, so with the lead in, the majority of the patients are above the 4 times PAIC90 and even towards the, if they're having a lead in, they're likely to be above the 8 times PAIC90, albeit at a lower level, a lower overall distribution of concentrations than that expected in non-pregnant individuals. So we're looking forward to the actual collection of this information of the data so we can confirm which prediction bears out. So next slide, please. We are also working with Craig Hendricks and Rachel Scott on a project to build basically a POP-PK meta-analysis across publicly funded studies for the dosing of oral tenofovir and emtricitabine. And so the data sets include both pregnant and non-pregnant patients taking oral tenofovir and emtricitabine. If we go to the next slide, and our key the next project ongoing is also looking at modifiable risk factors of pediatric delirium. So this is actually focused on developing PK/PD models to explore predictive variables related to the onset of delirium, and we're expecting the data to be ready for assessment actually this fall. And this will be looking at the link between the administration of midazolam, dexmedetomidine, fentanyl, and morphine, and the onset of delirium in this patient population. The next slide, please. And then I don't know, Sarah, if you wanted to take over for this one to highlight the education and engagement here.
Thanks, Rob. Yeah, so another key aspect of our pharmacometrics and clinical trial design core is education and engagement. And we've had representation at talks at the ASCPT 2022 annual meeting on using PK modeling in pregnancy. We will also be presenting a talk at ACOP highlighting the BPCA and Imprint Hub engagement of partners and work in enhancing maternal and pediatric therapeutics. Rob and I also have taken part in a number of workshops from the BPCA and the FDA, University of Maryland, pharmacokinetic evaluations of pregnancy and virtual public workshop there. That was also supported by Jeremiah Mumper and Brookie Best from the UCSD pharmacokinetics core from their CETs. We also have hosted webinars. Rob gave an excellent webinar kicking off our webinar series in December on model-informed drug development and discovery that you can access on the internet on our website at imprint.org. We are also planning some in-person and virtual training opportunities, and we are always looking for collaborations. I know Rob highlighted several that he's involved in, and I have several that we are in the process of developing, but are not at a stage where I can discuss yet. So with that, I will turn it over to Deborah Lynn, who will be speaking on behalf of Sean Granis and Christopher Martlow for our real world evidence core. Deborah?
Hi, everyone. So unfortunately, the original speaker, Dr. Granis, cannot be here today, had a last minute conflict. So I will be presenting the slides on their behalf. Next slide. The Imprint Real-World Evidence course sets out to create a collaborative consortium of real-world evidence for the Imprint Hub by providing an effective governance structure and share infrastructure for data sharing. The core will integrate and facilitate the secure and efficient access to diverse high-quality real-world data sets, as well as the informatics solutions that can support and accelerate clinical and translational research in maternal and pediatric pharmacotherapies and hopefully ultimately advancing the informatics innovation, knowledge, and best practices to accelerate the T2 to T4 maternal pediatric precision medicine. Next slide. So the core consists of two main data sharing sites. One is Nationwide Children's Hospital. The investigators leading this effort is Dr. Chris Bartlett and Dr. Carl Beggs. The other one is Indiana University with Dr. Sean Grenes and Dr. Colin Rogerson. NCH is located in Columbus, Ohio. It is one of the largest pediatric hospitals in the US with over 1.5 million patients visits per year. It has close affiliation with Ohio State University with many collaborative research on maternal and pediatric health. NCH cares for almost all of the NICU babies delivered at OSU Wexner Medical Center. So there are a lot of mother-infant pairs between OSU and NCH. In addition to the EHR data, NCH also has collected a rich data set on a small subset of patients through the Ohio Perinatal Research Network, OPRN. OPRN was founded in 2010 and collects clinical data on pregnancy outcomes, neonatal mobility and mortality, and childhood outcomes. The mothers consented to sharing EHR records as well as completing self-reports on health history. It currently contains around 1,900 mother baby diet as well as close to 1,000 fathers. Next slide. Since its inception in 2010, the OPRN has collected over 125,000 biological samples as listed here. In addition, it also collects neurodevelopmental outcomes up to 5 years of age. Next slide, please. So currently we are working on an NCH data linkage with OSU. Right now we have piloted a fuzzy lookup method for the self-linkage using Sundek's last name, first initial of the first name, gender and date of birth. The precision and recall are acceptable so far, so we're going to try it with the mother-infant linkage. And it is a little bit more complex, so we're still working on that. But we have developed a protocol for a decision tree method, and then I'm trying to find the optimal token for linkage. And if you're interested for more detail, please reach out to us afterwards. Next slide, please. So for the other data sharing side, Indiana University, there are two main resources. The first is the building blocks of pregnancy biobank, BBPB. It is a longitudinal perspective collected linked pregnancy outcome biobank within the Indiana University of School of Medicine Department of OBGYN. It started in 2009 with 3,200 participants and collected over 73,000 samples. Many participants have a complete set of samples in all trimesters, as well as labor and delivery. Notably, it has a good representation of Hispanic and Black patients. BBPB is a resource available for researchers focusing on pregnancy-related questions and topics such as preterm birth, gestational diabetes, and preeclampsia. The next is the IMPC, which represents the Health Information Exchange for the state of Indiana, receiving clinical data from over 100 separate healthcare entities in the state. The strength of the IMPC database includes encounter data, labs and pathology, orders, patient notes, and diagnosis data. It covers over 70 million patients with over 1.5 million clinical transactions, daily transactions. Patients in the IMPC are matched and given a local identifier so that a patient can be followed across multiple institutions. Next slide. So currently, Dr. Grener's team is applying a modified version of the phalanges center record linkage model, adapting it to maternal-child linkage. And they're validating the protocol currently with this linkage algorithm. Once the algorithm is trained, tested, and finalized, it will be applied to the full IMPC population. And then NCH at the same time will also run the algorithm on their combined data set for validation. Next slide. So the next step for the real-world evidence core is to continue to refine and test the protocol for mother-child linkage, as well as exploring the opportunities to enhance the IU and NCH data sets with social determinants of health. And finally, we want to establish the governance as well as the sharing, data sharing protocol so that data can be shared within the mPRINT network and participating sites. I think, yeah, that's about it for the real-world evidence core. Thank you.
At this time we can, we have about 20 minutes or so for questions and answers session. So feel free to raise your hand and or unmute or use the chat functions either in Whova or through Zoom, and we can follow up there. Aaron?
Yeah, can you hear me? I had to switch headphones. Yeah, so I've got a comment and a question for Sarah and Rob, and then a question for Lang. So, just sort of a comment in terms of the pharmacometric modeling. I mean, to me, this is not the only, but a great example of what we're talking about when we're viewing the mPRINT Hub as a marketplace and a resource, and something that can facilitate collaboration. And I was really appreciated the fact that, you know, already there are some collaborations going on in terms of PK models. So, you know, certainly, you know, the broad community, I would say a lot of us would very strongly say that would benefit from the use of pharmacometric modeling, especially as we start to really build in the physiological basis to these. So one, to the community out there, if you have needs or interest in this area, the mPRINT Hub is here to help you with that. So my question to you, Sarah and Rob, is, you know, with that context, in terms of, you know, there's certainly the repository will be built up, but what are your thoughts, or, you know, what are some additional ways that you could think about soliciting what would be the priorities for building those PK models within the community, and how can you facilitate collaborations in that area so that they can most effectively start influencing clinical trial design as soon as possible?
Sarah, did you want to take the lead on this?
Go ahead, Rob.
So
Welcome to
I
take
think
it.
it's an excellent question, because I mean, thus far it's been relatively relatively casual. People have reached out to us, or so actually the opportunity pool funding opportunities, the announcements have also drawn in specific inquiries. So we've had, actually, what we didn't show is there's probably what 8 or 10 more inquiries with respect to with respect to approaching these projects. So I think having some sort of maybe governance structure that can prioritize projects, that also maybe leverages what's already known and what's a relative, you know, what level of effort is required. Is it a new space where a completely new model will need to be developed versus something where we can effectively take something off the shelf? Can we leverage maybe personnel that are at the inquiring institution or the inquiring group to bring to equip them to actually apply some of these techniques and answer that question? And I know we've had these discussions in the steering committee meetings, in the PCTDC meetings as well. Perhaps even, you know, having a button on the website for specific inquiries. You know, here, for example, at UB in the CTSI, if you have certain questions, there's like not a screening form, but an intake form, for lack of a better term, that allows you to describe the project, and then it is reviewed and channeled to the appropriate individuals to maybe discuss further or give different levels of feedback. So, and I don't know if that helps to address what you're asking. And I don't know, Sarah, if you have some.
Yeah, so thanks, Rob. That was a great, and yes, on the Imprint Hub website, there's the ability either to email us at info@imprint.org, or fill out a form if you're interested in collaborating or gaining some insight from our pharmacometrics and clinical trial design core, or any of the other Imprint Hub cores. We'll screen those and send it to the appropriate cores. And I'll also note that this has actually been a discussion both from the BPCA initiatives, as well as the FDA Maryland CERC workshop on PK and pregnancy is prioritizing models and things beyond, obviously the examples that we're getting and the questions we're getting to support from investigators is really helpful. But then beyond that, looking at perhaps drugs that are metabolized through common pathways and trying to optimize PBPK models for those that then can be applied to other agents as well.
Hey, Sarah, if I may, I think trying to address Erin's question as well. I think Erin's question partially related to dissemination and engagement. I think we as a knowledge-based portal core, we actually had a very close communication with Sarah in a way. How do we disseminate the models in the knowledge base? It's actually a very tough question. Right now, the strategy we ended up with is, first of all, we wanted to record those PK model paper in the knowledge base as whether the PK model is available, right? Then we can cite back to the paper directly. And we have some general description of paper. However, the model parameter, that's a challenging part where every paper is different. Every model somehow is different. So we don't have I call it this kind of a ontology on a model yet. But I think in the next couple of years, we really want to get there, but it's just not there yet. So in the knowledge base, though, we do have PK data. If they're published through the non-compounded model, means that they're using the data. Summary stats talk about data, we have that, okay? We have the sampling time points, we have all those things in the knowledge base. However, the model itself, though, is not totally there yet. We're gonna say, this paper published this model, but we're not gonna be able to describe the model in knowledge base yet.
Great. No, I mean, I'm really glad to see that there's already collaborations and a queue coming up, and it sounds like a lot of thought into how to facilitate and prioritize. So I'm really glad to see that's happening. And I did have another question, but that could maybe wait till later, right? Because there is an interesting discussion popping up in the chat on the real world evidence and ER data.
So I think that's a power question I can answer as well. So in the Imprint, we do have a data sharing and modeling subgroup, working group. And as a matter of fact, in this afternoon, about 1:45, we're gonna talk about data sharing and a data usage agreement discussion in that working group. So technically for people outside of those institutes, they really can access, request the data. So we do have a master data use agreement development right now. Hopefully going to sign up by all the people in the network. So technically people can really request EHR data from, Imprint institutes.
Right. I see there's a question from Dr. Sticka about providing a concrete example on and how we envision this collaboration to work. And I'm assuming you're talking about the pharmacometric core collaborations. And we have some examples now where we've worked with networks such as Rob presented, working with the Impact Network to provide support for clinical trial design, as well as also support for then the analysis afterwards. You know, I think there's some funds within Imprint to help support the clinical trial design, and then, you know, building into, you know, hopefully some future grant funding to provide for the analyses or other sections of it that maybe we need additional support from where to provide funding for postdocs who actually do some of this work that's outside of our Imprint hub. There'll be, we're working on setting up some infrastructure and governances that would allow for that. The other examples we have are where, for instance, with our opportunity pool funding, where we are providing support for consultation and training for these. So offering hands-on ability for young investigators or fellows, for instance, to work with us to get experience in pharmacometric modeling or PBPK modeling and learn about it a little bit more and get the support through that method. Rob, I don't know if you want to add anything else to that.
No, I mean, I think that outlines things well. I mean, and actually a couple of those projects, this one, the emtricitabine ended up getting funded, and we ended up supporting the person who was supported on Imprint and is now primarily supported on that R21. We actually, you know, the vision is, you know, if we help in the initial phases, that if something is funded, that the continuing work is supported with that incoming funding stream.
I see the question from Sarah V from Vanderbilt asking whether there's a vision of linking the mom and the baby's data together. As a matter of fact, we do. Both OSU, Nationwide Children's Hospital and OSU Hospital, they are in the process of linking mom and the baby's data right now. It's partially is logistic. We have to go through the data sharing process between two hospitals. And Indiana University are going through the similar process. As a matter of fact, linking mom and baby's data is really an interesting research question as well, because, you know, we want to use all the information available through EHR to make the link as precise as possible. So there are ongoing efforts trying to make that happen, but we're not there yet. But this is definitely our vision, trying to link mom and baby's data into institute, OSU and IU side, you know.
Yeah, thanks, Lang. And I think this is really interesting. And we're, you know, at the Vanderbilt site, we have clinically, because we take care of moms and babies in the same system, it simplifies it a little bit. But I'm curious about once you have that linkage, you know, which baby or babies go with mom. And then there's the time element, making sure you link the right pregnancy at the right time to the mom if she has multiple children in the system. But then is the vision to take like all of the electronic health records and like try to share that into some sort of centralized database that will then update over time? Or is the idea to extract certain variables out and sort of create some meta tables of, say, drug exposures and important outcomes? And, you know, because those are two very different scopes and require sort of different planning for us as we think about how we could use this resource in the future.
Agree. I would say if it is within the same institute, saying, you know, like Ohio State, I think that the vision here locally is really having a integrated mom, you know, baby's data all together. So we do have ongoing so-called data lake, institutional infrastructure to do it. Then IU has similar thing as well. For many years, our registry been doing this already. However, across multiple institutes, we have to rely on the master data user agreement to share the data. So that's the way we wanted to address this question challenge.
I think I can add to that a little bit. I think that the goal initially at IU, at least, is to create a master set of data. It's not going to be linking the entire EHR set between mothers and babies and making it available. It will make a registry of certain fields available, and we're still developing what those may look like or what's available through the majority of our INPC data systems, our health that are linked through Regenstrief. I think the goal is to create a registry that we can then share openly with fairly easily with investigators that will control, but then there will also be the opportunity if we haven't captured the field that you're really most interested in, or it may require some natural language processing to go through the unstructured data, then we could go back to the data core at Regenstrief to pull separate. That may require an additional charge from what we'd make available through the Imprint Hub.
Thank you.
Aaron has more questions. Aaron, go ahead.
Yeah, so actually, I saw a couple of questions in the chat. One from James Antoon and another from Mike Liebman, and it interfaces with a question I had, and I think some of the discussion that just came up. Yeah, so one of the challenges, certainly, I think one of the challenges to get to incredibly large data sets and combined data sets comes down to, harmonization, common data elements. And one of the questions I had for you, Lang, and this might be better tomorrow at the hour focus, in terms of creating an ontology, and how does that map to existing disease ontologies or things like the monarch ontology. So maybe if you just comment in terms of how we can what could be done to address common data elements, differences in terms between different universities, and how this can start to all map together. Or again, this I think is a prioritization thing, because obviously we can't solve this for everything, but
So this is a really loaded question. I want to talk a little bit from two aspects. One is really EHR data. I think that there are two practices, I would say. In general, from physician standpoint of view, physician get used to ICD-9 code and the Loniger code and some type of medication data as well, right? So from that angle, I feel sometimes for collaboration, building up the collaboration, using those established ICD-9 code, like Loniger test, the medication will be a common way to do it, okay? And it's in the EHR system. As a matter of fact, I would say almost every EHR system we have so far is Epic. I think Indiana has a little bit more exception as Cerner, but nevertheless, people are still using a similar thing. Okay, so that's one thing from physician clinical standpoint of the collaboration. It's not a major challenge. From people doing informatics, on the other hand, people using other terminology, for example, OMAP terminology, where transform all those data into OMAP format, where people can do large scale computing on top of that. That largely is also available through the IT, kind of a CTSA infrastructure from Multi Institute. People use OMAP data formatted to translate the data points in OMAP format. So somehow that's also available as well. So that's not a major issue in terms of data sharing. The major issue is all about clinical notes. There are a number of, for example, adverse drug events that are largely disputed, not only in the structured data, but also clinical notes. That's where the clinical phenotyping is a lot of delivery across different institutes. So that's something I think is going to be collaborative effort. Is also going to be disease dependent as well. And data sharing has also challenged because clinical notes typically are identify the data, right? So the data itself cannot really be shared. But I think de-identified data after phenotyping algorithm generated, those data can be shared, but needs to be validated across different institutes. So that's needed quite a bit more collaboration. So that's how I can say about this data sharing from EHR data. On the other hand, the knowledge base, a protocol knowledge base, we are building up the knowledge base using existing terminology. I think what we're trying to accomplish is really using existing terminologies like ICD-9, LONIC, and maybe medication data as well, to map the data in the knowledge base to the standard language. And we're not there yet. We just started, and we feel there's a lot of diversity, especially clinical endpoints. There's a lot of diversity over there. But fortunately for PK part, it's pretty straightforward. We have done a lot of work on that. So there are low-hanging fruit in the knowledge base, kind of ontology terminology developments. There's also more challenge in terms of how do we normalize the clinical data. I think that's also the area we wanted to collaborate with from GKB as well, because that's the things from GKB has done, for more than 10 years already. So that could be something we can work with them and trying to figure out a strategy between us and them. I hope I answered the question.
No, yeah, a lot to do, and I'm really glad you're thinking about ways to get there and areas that we can demonstrate what can be done, because I think it's going to be important to prioritize. Obviously, can't harmonize all of EHRs in one go.
Are there other questions from the audience? Or suggestions on how we can prioritize our work ahead? That's really going to be our focus on tomorrow afternoon session is working through some of those prioritization goals.
There's some additional comments in the chat. Sharon Nachman from Impact asked if the TriNet X database can be of any help to investigators. I don't know, Lang, if you're familiar with that. Sarah, this seems like it's a real world data effort. And then Michael Liebman asked about ICD-9, ICD-10 codes being developed to support reimbursement business practices. They do not really address the true clinical view of the patient from the clinician's perspective. So my guess is you're asking how do you reconcile that when you're preparing a
let me kind of expand on Michael's question about ICD-9, ICD-10 codes. Okay, so technically, data integration and a meaningful clinical phenotype definitions are two different concepts. Okay, so from data integration standpoint of view, ICD-9 code, ICD-10 code is a great standard because everybody using the same thing. So we can easily integrate, Vanderbilt data to IU data, OSU data together, right? So that's easy. That's the things we wanted to do. However, once you get in ICD-9 code, how you define the disease status, that's a tougher question. And it needs not only ICD-9 codes, maybe the other unstructured data as well. So that takes a lot more effort trying to make sure how those data be utilized for the clinical purpose. So to me, again, ICD-9 code, ICD-10 code is a great resource for data integration, but not necessarily adequate resource for clinical usage. Okay.
I agree. There's also issues in looking at maternal and health, especially in identifying the length of the pregnancy as well. Even worse is identifying lactation in electronic health records, because those are things that are not recorded. So there's a lot of issues with some of the real world data that we'll need to come over and come up with new information on. So.
The question about from James Anton about Centennial. The answer is absolutely yes. I think claims data historically has reasonably well organized against kind of ICD-9 code. I think this is where we really wanted to get an opportunity to work with Centennial collaborate as well.
Yeah. One of the issues there is that the FDA Sentinel database uses their own dictionary, and whereas I think all the imprint hub centers currently use OMOP. So we'd have to come up with a way to link the OMOP to the FDA Sentinel dictionaries as well. But definitely is on our radar. So. Great. So I think we've come to the end of our session on the DMKRCC. Definitely, if you have other questions, continue to ask them in chat or send us emails or communicate through the Whova app. Again, there will be the talk tomorrow with Lang on the knowledge base, and I'm sure more of our real world evidence questions will be addressed during the next session with the phenotyping core at Vanderbilt University. So now I'm going to turn the talks over to my colleagues at Vanderbilt.